Okay, on mutant selection. Now, because mutation is a rare event, even in bacteria, we must have robust methods of identifying or selecting these mutants. If you remember, mutants are produced either via spontaneous mutations or by induced mutations. Induced mutations happen when we subject the cells to factors such as chemicals or things like radiation, and mutation happens in the DNA. So the next question would be, how would we selectively identify or differentiate these mutants from the original parental wild type cells, right? So there are basically two ways that we do this, two main approaches. Um, the first method is direct selection. Very simple, very easy to do, sort of an experiment. So what happens is you would inoculate the cells onto medium that would support the growth of the mutant, but the parental cells will not grow. Only the mutant cells will grow. So for example, if you look at this diagram, so let's say you have bacterial cells that are resistant to streptomycin, which is an antibiotic. So you have a mixture of cells that are resistant to the antibiotic, which are the mutant cells, and you have cells that are susceptible or sensitive to the antibiotic, which are the wild type cells. So now you need to identify, you need to pick out these mutant cells, so that you can subject them to other studies. So what you could simply do is prepare a medium, prepare an agar plate like this. It could be standard nutrient agar, or depending on the growth requirements of the bacteria, it could be maybe a TSA plate or whatever agar that supports the growth of that bacteria. So you prepare a plate of that appropriate agar, and that plate should also contain the antibiotic that the bacteria is um, resistant to. So in this case, because we want to isolate bacteria that are resistant to streptomycin, then we would prepare an agar plate that contains streptomycin, right? So, and of course, you must also have a plate that doesn't have streptomycin, which serves as a control. So, on on this plate, on plate A, where you have the antibiotic, typically the parental wild type cells should not grow, right? Because the wild type cells are sensitive to streptomycin, right? They are susceptible to streptomycin. Streptomycin will kill them. The only cells that can grow on this plate are cells that are resistant to streptomycin. So any colonies that you see on this plate are basically indicative of the mutant cells that are resistant to streptomycin, right? So very simple thing to do. If you want to identify cells that are resistant to maybe ampicillin, then prepare an agar plate and add the appropriate concentration of ampicillin, and you spread the cells on the plate. The colonies that you see are basically from cells that are resistant to ampicillin. So this is called direct selection. Of course, you must also have a control plate that does not contain the antibiotic. All right. And on this plate, you should see a lot more colonies, because this is a normal plate. This could be your normal nutrient agar or TSA plate. So let's call this plate B. So this plate doesn't have the antibiotics, so pretty much all the cells can grow on this. Mutant as well as non-mutant cells can grow on this plate. So naturally, you will see a lot more colonies. Now, the purpose of this plate is it serves as a control, because in the event that you do not see any colonies on this plate, right? If let's say you do not see any colonies on plate A, there are two possibilities. Either Possibility 1 is that you have no mutants. Possibility 2 is that you have no cells at all. Right? So these are the two possibilities that will result in you not seeing any colonies in A. So if you have a control plate, which is plate B, that doesn't contain the antibiotic, and if you see colonies on this plate, that will eliminate option number 2, which means you do have cells in your test tube, right? It's just that you do not see any colonies on plate A because there are no mutants. So your plate B serves as a control plate. This is called direct selection. Very simple thing to do. Now, the next method that we can do is indirect selection. Now, in indirect selection, again, you want to isolate the auxotroph, which is the mutant, from the prototroph, the parental wild type. Now, this is a little bit more challenging because unlike your direct selection method, in this method, whichever media that supports the growth of your mutant, it will also support the growth of the or the prototroph. Because remember, prototrophs are the wild type cells that do not require any additional growth factors. They basically produce everything on their own, right? Or you do not need to add anything else to that media. But in the case of auxotroph, these are mutant cells that lack the ability to produce at least one or more growth factors. So these growth factors must be supplemented into the growth media. So if you have a plate that has been supplemented with growth media, then the auxotroph will grow and the prototroph will also grow, because the prototroph doesn't need the additional growth factors. It makes its own, so it can still grow on such a plate. So this is a little bit more challenging, and for this we use a technique called replica plating. Now, replica plating is a technique that has been around for a very long time. It was described in great detail in a paper published in 1951 on how this technique is very useful in identifying auxotrophs. So what happens is, so you would prepare a master plate, which is usually your standard nutrient agar, okay? And this is your plate. And of course, because this is your nutrient agar, the auxotrophs will grow and the prototrophs will also grow. Okay, so as you can see, this plate has a line that has been drawn on the wall of the petri dish. Okay, next you prepare another plate like this, and you cover the plate with velvet, which is a type of fabric. It's a little rough in texture, and a similar line is also drawn on this second plate. So you take the first plate and you gently press onto the second plate and make sure that the line on the first plate and the line on the second plate, they align. So when you do that, it means the cells from plate 1 would be transferred onto the exact same location on plate 2. All right. So the cells go on here, this cell will go on here, so on and so forth. So if you look at the imprint pattern on the second plate, it is basically identical to the colonies that are seen on the master plate. So you have cells here that came from this colony. You have cells here that came from that colony. And the cells here, 1, 2, came from this 1, 2. So so the cells have been transferred from the master plate onto this fabric. It's a piece of cloth, right? Velvet. Next, this plate, the plate with the imprinted colonies, will now be your central plate. Next, you would pick up a sterile nutrient agar plate. This is your standard nutrient agar, similar to your master plate. It's a clean plate, and you would also prepare a glucose salt agar plate. Now, this agar plate is special in the sense that it only contains glucose and some salts. All right, so which means only cells that can make use of glucose and salts can grow on this plate, because every other nutrient is absent. On your nutrient agar, right, this nutrient agar contains all the essential nutrients such that your auxotrophs, the mutants, can grow, the prototrophs, the wild type, can grow, because all the nutrients are present here. On glucose agar plate, only the prototrophs can grow. The mutants can't grow because the mutants require additional factors to be added into the growth media. But on this plate, the agar contains only glucose and salts. So the prototrophs can grow because the prototrophs can make use of the glucose to produce all the necessary growth factors and nutrients. But the auxotrophs cannot grow on this plate. Okay, so take the plate. All right, take both the plates. First, take the nutrient agar plate, gently press it onto this velvet. All right, and what happens? You will have some cells that will be transferred from the velvet onto the agar, right, in the exact same locations as you can see here. Okay, this is the product of it. So the colony that you see here is the colony here, and the colony that you see there is this colony here. So and of course remember that you must draw a line on the Petri plates, and when you press the plate onto the agar, you have to make sure that the lines align. So you transfer the colonies, you transfer bacterial cells onto the nutrient agar, you incubate it, and 24 or 48 hours later, you would find colonies appearing on the plate. The same thing, you take your glucose salt agar and gently press it on the velvet. The cells would be transferred onto the agar. Likewise, you incubate it for 24 to 48 hours. And colonies would appear. So you compare the position of the colonies between these two plates. Okay, so the colony that is present here is also present here. The colony that is present here is also present here. So the colonies that are present on both plates are basically the prototrophs, right? The prototroph can grow here, and it can also grow here. Okay, but the colony that grows on this plate, okay, I'm going to call this plate A, on this plate but doesn't grow on plate B, has to be the auxotroph, because remember, on glucose salt agar, auxotrophs cannot grow. So if you look at this particular colony here, sorry, okay, so if you look at this particular colony here, you find it on the nutrient agar, but that colony is missing on this agar plate. So which means that is the colony that contains the auxotroph, because here prototrophs and auxotrophs can grow, but here the auxotroph doesn't grow. So these are the mutant cells that you want to pick and analyze. Right, so as you can see, this particular method, which is called replica plating, because you make replicates of the plates, It's a little bit more tedious than direct selection, but it is a very useful way, a very good method of picking up your auxotrophs from the to differentiate your auxotrophs from the prototrophs. But again, remember that this works on the principle that we are using two different types of media. We are using nutrient agar and glucose salt agar. Nutrient agar contains all the necessary nutrients such that the auxotrophs as well as prototrophs will survive. Glucose salt agar contains only glucose and salt. So only cells that can make use of the glucose to produce all its essential factors, all its essential nutrients, amino acids, and everything else, only such cells can grow here. And that would be your prototrophs, right? Your auxotrophs, which are mutant, will not be able to grow on glucose salt agar because they are mutant and they lack the ability to produce one or more growth factors. So those growth factors are supposed to be supplemented. So the auxotroph will not be able to grow on the glucose salt agar. And then you compare the position of the colonies between these two plates. And it's very important that the lines be drawn on the plates and that the lines are aligned so that you can compare the plates. If the lines are not aligned, then you wouldn't be able to compare the colony for colony.